Welcome back to my course Industrial Biotechnology. Now, in the last lecture, I tried to cover the biodiesel, which is appeared to be the one of the promising bioenergy source for the future. And I told you that the biodiesel can be produced from the vegetable source, from the animal fat. Also, it can be produced from microalgae. And basically we extract this lipid or fats from this sources, and then it undergoes the esterification process. In presence of methyl alcohol and acid catalyst, it produces biodiesel, and which is nothing but methyl esters of the higher fatty acids. Now, today I want to discuss another promising biofuel for the future, that is the bioethanol. And this butanol can be produced both through the biological process as well as chemical process. We will be talking about how this butanol production take place through the fermentation process. And here I want to add some history behind. We'll discuss the detailed history on the butanol production process, and particularly the importance of the butanol production that has been given most stress during the World War II, because that time there was the acute shortage of crude petroleum. So USA was looking for some kind of substitute for this petroleum. So they considered this butanol as because it has good calorific value, and also it also has higher seated number, which is very much required for running the vehicles. Now, if you look at the butanol, it is a kind of higher alcohol, and butanol is an alcohol that can be used for transport fuel. I told you it is used there, and I told you when I discussed the ethanol fermentation process, then also I discussed that with ethanol we produce some kind of that byproduct as the higher alcohol, what you call fusel oil, and this is also contains the butanol, amyl alcohols and other alcohols that we have, and it is a higher member series of straight chain alcohol with each molecules of butanol. Say it is C4 carbons, that 4 carbon ring that we have, carbon chain that we have containing the 4 carbon atoms. It is largely produced by petrochemical industry, used as a chemical and solvent. It is miscible with most of the solvent like alcohol, ether, aldehyde, ketones and aliphatic anaerobic hydrocarbon. Is sparingly soluble in water. Water solubility is only 6.3% and is highly refractive compounds. Now, chemically it looks like this. This is the 1 carbon, 2 carbon, 3 carbon and 4 carbon, and this is the wedge group at the end. We have 1 butanol, we have 2 butanol, we have isobutanol, we have tertiary butanol that we have. Now, if you look at the uses of ethanol, it can be used as a lacquer. Then it is used as for the production of this photographic film, and metal cleaner and solvent. It is and drug extract and dehydrator and synthetic resin and plasticizer, and also dry cleaners. So these are the different things that we have. Now, here we have given some kind of information that world butanol demand that we have. This is total is 1.3 billion gallons, and where it is used, that has been noted here. We can see most of the butanol used for acrylate production, and then we have acetate, then we have glycol, ethers, then solvent and plasticizer. Now, use of butanol, here it is clear that why butanol used as an energy source, because butanol the energy density is 10 to 20% lower than gasoline, but it is higher than ethanol. But you can see the gasoline has the calorific value 32 megajoule per liter, whereas butanol is 29.2 megajoule per liter, which is very close to gasoline. But if you look at the ethanol, as compared to ethanol, the calorific value is quite high, and also as compared to methanol, this also butanol has higher calorific value. The another advantage is that it has lower vapor pressure, lower volatility, and evaporative emission, which is very much required for the fuels. And increased energy security can be produced domestically from the varieties of feedstock that I discussed. Different organic matter can be used for the production of butanol. Butanol is oxygen tolerant, so it can mix easily with gasoline or diesel. Fewer emission carbon dioxide captured by growing the feedstock reduces overall greenhouse emission due to this use of butanol. Now, butanol, we have make a comparative study how the characteristics of this differ from that of other fuel like gasoline, ethanol, and methanol. We have energy density here is, as I pointed out, it is very close to gasoline. Then air fuel ratio, that is also close to gasoline, and heat of vaporization, that is also close to gasoline. If you compare with ethanol and methanol, that heat of vaporization is quite high. And octane number, this is also very close to gasoline, but in case of obviously ethanol, we have octane number is quite high, and motor octane number, this is also very close to butanol, is very close to gasoline. Now, history of butanol production, that is the production of butanol by the biological means was first performed by Louis Pasteur in the year 1861. 1905, the Austrian biochemist French Skardinger found that acetone could be similarly produced, and 1910, then Farbeck, they developed a bacterial fermentation process using the potato starch as a feedstock in the production of butanol. Now, butanol production through the fermentation process, one scientist plays very important role, whose name is Weizmann. He isolated the Clostridium acidobutylicum. It is the obligated anaerobe, and he patented. He got the American patent, which is because it has the capability to produce high concentration of butanol. After Second World War II, acetone butanol fermentation becomes non-profitable as compared to the production of same from the three solvent like acetone, butanol, ethanol from as compared to that of petroleum, because acetone butanol fermentation process, because butanol is produced through the acetone butanol fermentation process, where it produces not only the butanol but also it produces acetone as well as ethanol. So that is the byproduct of that particular industry. So that is one of the major problems that we have. Now, disadvantage of biobutanol is several. The production of biobutanol is quite low. The concentration, if the I already mentioned you that if the concentration is low, then the recovery cost will be very high. The cost of the product depends on the concentration of the particular product present in the fermentation broth. So we find that then through the fermentation broth nowadays, that is reported in the literature, we got around 4 to 4.5 grams per liter, which is quite less. Now, biobutanol may cause the gas gauge reading mistake in vehicles. Biobutanol may yield more greenhouse gas emission per unit motive energy extracted compared to bioethanol due to the less octane number. So, these are only the high viscosity of biobutanol may lead to the potential corrosive and aggradation problem, when it was used in spark ignited engines. So this is the biobutanol market. We can see the market that we have in Asia, except China it is 13%, Europe 25%, North America it is 24%. This China is itself has very high market, that is 34%, rest of the world is 4%. And this is the pattern of use of bioethanol or production of butanol that is showing with respect to time how it is increases. Now, as I pointed out before, the butanol can be produced by two ways. One is chemical means, another is biological means. Now, biological means it is produced, I told you it is a acetone butanol fermentation, where we get three solvent. One is acetone, another is butanol, another is ethanol. Now, in case of chemical process is used from the petrochemical industry, and the microorganism that is largely used for biobutanol production is the Clostridium acidobutylicum and Clostridium bijarinki. This is the two organisms largely used, and also we have Clostridium coniboi that is used in Japan. They are obligate, as I told you, obligate. Here I want to point out that obligate anaerobe is a little bit difficult to handle, because trace amount of oxygen present in the fermentation broth will kill the microorganisms. So, totally the dissolved oxygen concentration of the fermentation broth is you have to remove, then and only then your organism can grow in the fermentation media. This is the only disadvantage we have. Another problem with anaerobic fermentation process is that rate of reaction is quite slow as compared to aerobic fermentation process. Now, selection of microorganisms, that is very important. I have already shown before also that microorganism plays very important role. If you look at that Clostridium acidobutylicum can ferment the starchy raw material like corn, rice and wheat at 37 degrees centigrade. Clostridium saccharoacidobutylicum and Clostridium caniboi can ferment molasses, saccharides and starchy raw materials. Clostridium saccharoacidobutylicum can ferment at 30 degrees centigrade, and Clostridium coniboi at 37 degrees centigrade. Bacillus tetrail, this is can ferment the inverse sugar portion of molasses. Optimum temperature is 35 degrees centigrade. Now, acetone butanol fermentation process, that I told you three solvent we get in the fermentation broth. One is butanol, another acetone and ethanol. If you look at the ratio, that how much is present there in the fermentation broth is 6:3:1. The process also makes the recovery amount of hydrogen, because this we observed, because this has three different phases. One is called acid formation phase, another is solvent formation phase, what we call solvent genesis, and again it produces the acid. So it had three different phases. In the initial phase, in the acidogenic phase, the acid formation take place, and we observe very good amount of hydrogen production that take place in this particular phase. So, so we can collect the hydrogen and use as a source of energy, and simultaneously we will be getting the organic acid. Then in the second phase, this organic acid will be converted to butanol, acetone and ethanol. Now, this is the metabolic pathways, how this formation take place. We have acetate, butyrate, and this forms, and this also forms when it is passed through the solvent genesis process, it produces the ethanol, acetone and butanol. So, it has been shown here that this is the Embden-Mayer pathway through which we get the acetyl-CoA, and this acetyl-CoA, from acetyl-CoA we can get the butyric acid, as well as we get butanol, we get acetate from, and also this side we get acetone and ethanol. This is the important metabolic pathway for one butanol production process, also given here how it is produced, butanol. So, what we want to point out, that that after pyruvic acid it produces acetate as also ethanol. These two process is to be closed. You have to inhibit this process, then and only then you can have the accumulation of butanol in your fermentation broth. The steps involved in the butanol fermentation is several. We have feedstock, then we have upstream processing, then we have acetone butanol fermentation process, then we have the fermentation products, and then we can see some butanol usage. Now, if you look at the feedstock, it is very interesting. It gives the different sources of raw materials, agriculture residue or byproduct of the crop biomass, non-edible crop biomass, wood-based biomass, industrial byproducts, municipal solid or biodegradable waste. Then we have sustainable. This is the sustainable indicator, economic, environmental and social indicators. So, that is then the upstream processing. We have pre-treatment process, we have biological, chemical, physical and physicochemicals. Now, I can give a typical example that if you use the kind of cellulosic material as a raw materials for this fermentation process, then you have to go for either acid hydrolysis or alkali hydrolysis, or you have to go the biological that enzymatic hydrolysis process. So, just to convert the cellulose to the glucose, and also you can have the in process you can have the physical and physicochemical system. Physical system means you can cut the size of the solid particle into small size, so that your bacteria or enzyme can act more with the raw materials. The hydrolysis and dilute acid and concentrated acid that is used for this hydrolysis process, and detoxification, that is the adsorption and enzymatic evaporation, extraction, ion exchange and resin and overlapping. This is the process we can do for the detoxification of this particular residue that we get from this. Then this affects the economy of the process obviously to a great extent. Now, acetone butanol for fermentation process can be carried out both by batch process, fed batch process and continuous extractive flash, two-stage immobilized cells. So, different processes we have simultaneous saccharification and fermentation, that where both the saccharification your organism that used for the saccharification of the starch or cellulosic material, and also it produces also the butanol. So, usually we consider kind of mixed microflora for doing all this thing, use of E. coli or other microorganism that is used for this process. Now, fermentation product, we have major products, we have acetone, butanol and ethanol, and then this is separated out through the distillation process, and then this can be used as a transport fuel, used as solvent I mentioned before. Now, if you look at butanol fermentation process from lignocellulosic material needs the pre-treatment I told you. This is enzymatic hydrolysis, we get the glucose, we separate the lignin, then sugar, it converted to acetone butanol, and this is the separation process. We separate the cell mass and we solvent recovery through the distillation process, we get the butanol. Now, this is the block flow diagram, and the block flow diagram for the production of butanol from corn starch. Corn we take out and we dry corn, we do the milling. Milling means it kinds of we are making it powder form. Then we make a dough and undergo the saccharification process, then the soluble material containing the glucose that undergo the fermentation process, then we have degas, then water removal, dehydration, acetone removal and the butanol, ethanol removal. That here this glucose whatever is produced, that is converted to acetone, butanol and ethanol, that slowly we can purify it, and finally we get the normal butanol. Now, raw materials used, as I mentioned before, that you can use the corn concentration is 6 to 8%, molasses concentration 5 to 6%, lignocellulosic material, the research, this is in the research level. Deficiency of phosphate and nitrogen in molasses is overcome by adding the calcium hydrogen phosphate or superphosphate. The concentration of ammonium salt is about 0.4%, and P2O5 phosphorus pentoxide is about phosphorus in the form of phosphorus pentoxide is 0.14%. Clostridium acetobutylicum contains the weaker cells and higher yielding spores. The higher temperature destroys the spores and heat shock measured to active the culture. This is very important step we find when we try to use the Clostridium acetobutylicum, then we find the heat shock is very much required just to activate the culture. Now, pH of the fermentation process is 5.5 to 6.5. pH changes from 5 to 7 during the fermentation process. The carbon dioxide is obtained anaerobic condition. Time of fermentation is 2 to 3 days depending on the culture condition and strain. The yield is like this, 30 to 33% on the basis of fermentable sugar and 25 to 26% on the basis of fermentable corn. Now, here we have very interesting things that we have I told you it this acetone butanol fermentation take place in the 3 different phases. This is we consider phase 1, this considered phase 2, and this considered phase 3. This is time, and this is your acid concentration. So here the acid formation increases and then it decreases. When it decreases, then you have more accumulation of solvent, again finally it is increases. So usually we carry out the fermentation up to this level, because this is the level cannot to have more solvent accumulation in the fermentation broth. So this is the you can see in this figure, this is the butanol concentration is quite high, then acetone, then ethanol, and this ratio I mentioned before, 6:3:1. And you know that I told you during the acid fermentation and acid production process, the hydrogen gas is produced along with the carbon dioxide. This is the total gas. This hydrogen gas we can tap and use as a energy source. This is the profile of different acid. This is butyric acid profile, this is acetic acid profile, how it is changes with respect to time in the acetone butanol fermentation process. Now, contamination during the butanol fermentation process is 2 main type, one is bacterial and bacteriophage. Now, here I want to point out that in the bacterial fermentation process, that only that some other bacteria, there is every possibility some other bacteria also may interfere in the process, but one advantage of this process is that this is the obligatory anaerobe. So obligatory anaerobe conditions that a few organism can grow, but anyhow that we face lot of problem with the other bacteria, because lot of other couple of bacteria can, particularly I can give the example of methanogens, they also obligatory anaerobe, they also can grow here, but only the difference is that here the pH is quite low, so methanogens will not be problem, contamination problem never arises, but in case of maybe the other acid producing bacteria may contaminate in this process. But bacteriophage is a common problem for bacterial culture, because bacteriophage is present there. I told you when I discussed the cheese making industry, that is one important problem that we have with the cheese making industry, that bacteriophage contamination problem. The bacteriophage is kind of virus, they attack a particular bacteria and they kill the bacteria. That is the problem, and that is why I told you, you can remember that cheese making industry, usually we preferred the mixed culture, so that even one bacteria is killed, other bacteria will keep on functioning. So that is one of the way we can recover this kind of problem. But here, since we are using the pure culture, if the bacterial phage is contamination, the whole process will be affected. Now, bacterial contamination, timely sampling of the culture from time to time, usual contamination is the lactic acid bacteria. Lactic acid contamination can be controlled by sterilizing the pipe fermenter, etc. before use. Now, this is the bacteriophage, how it attack the bacteria, how it attacks the bacteria. The contamination of bacteriophage contamination cannot be seen by the ordinary, cannot be seen by ordinary microscope. This is because virus is a very small particle, so we cannot visualize under the microscope. It is much smaller than bacterial cells. We know the size of the bacteria is 0.5 to 2 microns, so it is much less than that, so it is very difficult to visualize in the ordinary microscope. So this is the problem, that is bacteriophage can attack the bacterial cell DNA, but the viruses can grow on the living cells, and operation is immediately terminated when the phage contamination is detected. That is the problem that we have. Now, we have downstream processing of butanol. Butanol recovery from the can be challenging in terms of energy, as this concentration is very low, that 20 gram per liter, and it is a high boiling point at 170 degrees centigrade. So distillation is mostly widely used in this method till death. So in conclusion, I want to tell you that butanol is considered as a promising fuel bioenergy source for the future, because if you look at the calorific value of butanol is very close to gasoline, and not only that, we find the Seaton number is also close to gasoline, and vaporization of this particular butanol is comparatively less as compared to ethanol. So it is more promising as compared to the ethanol. Because I told you that ethanol can be used also as a power alcohol, and Government of India or throughout the world, it has been found that if we replace the gasoline 10 to 20% by ethanol, the same engine configuration can be used for running the vehicles. So similarly, but here the advantage is that butanol, the calorific value is close to gasoline, so we don't have any the energy problem, energy conversion problem. But when we use the ethanol, the energy content will be, the combustible energy is quite less as compared to butanol. This is the problem that we had, and butanol biobutanol can be produced through the biological, the different processes, but this process is obligatory anaerobic fermentation process, where this anaerobic fermenter, only the obligatory anaerobe then can grow. Examples are Clostridium acidobutyllicum, and other bacteria also use. And I told you one important feature of this particular organism is required heat shock to activate the culture. We also carry out this experiment in our lab, and we find that unless we do the heat shock, this organism will not grow properly. So then, and usually that butanol is recovered through the distillation process. Only the problem is that it is the higher boiling point, 117 degrees centigrade. So we can collect on the basis of the fraction of the distillation. So we in the fermentation broth, we get the 3 different solvent in the of butanol, acetone, and ethanol in the proportion of 6:3:1. So I think it is all about the butanol fermentation process. In the next lecture, I shall cover one very interesting topic that is largely in operation in our country, that is the biofertilizer, and it has the great prospects as per India and abroad is concerned. So thank you very much.